KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, so in this video now we are going to discuss a new chapter. New chapter name is Biotechnology Principles and Processes. So in this particular case, so definitely we will learn information regarding principles as well as processes under biotechnology chapter. So let us have a look on this chapter once. So new chapter name is Biotechnology Principles and Processes. Biotechnology Principles and Processes. So in this particular case, so first point, what is the point now we are learning now? For the first time, Carl Erike, this is scientist name, Carl Erike used the term biotechnology. One more time. For the first time, the term biotechnology was used by Carl Erike. Now, biotechnology deals with the techniques, so of using living organisms as well as the enzymes and from the organisms. So means here definitely we are using the organisms or the enzymes for what? For the producing of products. So for the producing of products we are taking. So live organisms or the enzymes here we are taking. So thereby what we are going to produce means we are producing different types of products and processes useful to human beings. Means in this particular case now in biotechnology chapter we are going to produce products as well as we are going to follow the some kind of processes. So by using of some kind of processes definitely we will get the benefits. So likewise, so biotechnology deals with the techniques using of live organisms or enzymes from the organisms to produce products as well as processes useful to human beings. So this point I hope you understood this. Next one, in this sense. So in this sense, so what are the things we can be taken means making of curd, bread and one more wine. So which are all microbe mediated processes could also be taught as a form of biotechnology. So means in this particular case now what is the thing? So in our home definitely we will prepare, mummy will prepare curd from the milk. And likewise in a bakery bread preparation we will be observing. Next one, wine preparation we will be observing in beverages industries now also there. So all these kind of processes definitely microbes mediated. Why? Because here for the production of or for the making of curd definitely lactobacillus bacteria is required, and for the bread preparation point of view, which one is required? Saccharomyces cerevisiae yeast is required. Next one, likewise for wine preparation also definitely yeast only is required. So likewise in this particular case, this entire processes we will be taking as microbe mediated. Means suppose in absence of microbes, so these kind of processes will not be completed. So such kind of processes so could also be taught a form of biotechnology. Means here these kind of processes means when we are going to follow these kind of microbes mediated processes, these kind of processes also we can be taken as which one? Biotechnology. Next one, furtherly many processes or the techniques are also included under biotechnology. So means these are all, first of all, three processes we have seen now: making of curd, and the preparation of bread and the production of wine. Likewise we have seen now not only all these things, so furtherly many processes and techniques are there. There are many processes as well as techniques are there, so which are included under biotechnology. So remaining further processes and techniques also we can be taken so under the biotechnology. So what are those? So for example, in vitro fertilization. So in vitro fertilization leading to leading to the formation of test tube baby. So test tube baby and synthesizing a gene and using it developing a DNA vaccine. So formation or the making of or the creation of or the discovery of a DNA vaccine correcting a defective gene. Next, correcting of a defective gene means gene therapy. In gene therapy we will use now. So likewise, so next one and are the parts of the biotechnology means here microbes mediated processes we have seen. Next one furtherly we are observing there are different types of processes and techniques are there under biotechnology. What are those? In vitro fertilization leading to the formation of test tube baby we are taking. Next one is synthesizing a gene, synthesis of genes we are taking or we are doing. Next one using it developing a DNA vaccine. So here synthesizing a gene means here we are trying to synthesize or we are synthesizing a gene. So by using of that particular synthesized gene we are developing DNA vaccine. Not only these two, one more also is there, correcting of a defective gene. So what is a defective gene is there? No. So that particular defective gene, by removing of that defective gene, so we in the place of a defective gene we can introduce normal gene. So thereby that particular organism function will be normal. So likewise, so defective gene we can correct and DNA vaccines we can be produced. Next one, test tube baby also we can be produced by in vitro fertilization. Likewise, all these kind of processes we can be taken as biotechnology. So all these technologies are the part of biotechnology. So definitely part of biotechnology. Next one, EFB is nothing but European Federation of Biotechnology. EFB is nothing but European Federation of Biotechnology. So EFB has given a definition of biotechnology. So it has given a definition of biotechnology, that is encompasses both the traditional view and modern molecular biotechnology. So the combinations of which one encompasses of which one and which one? Traditional view as well as modern molecular biotechnology. So based on this one, now they are going to give one definition to us. So that definition is, so now here you see definition of the EFB is integration of natural science and organisms, cells and parts thereof and molecular analogues for products and services. Services SCR, you know. Now here this is the exact definition of which one? EFB. One more time, integration of natural science. So this integration of natural science with whom? Organisms and within the organisms cells, in cells parts of the cells and what are those? Molecular analogues. So all these things for what? For producing of products and for service, for giving of service concern. So likewise they have given one important definition by European Federation of Biotechnology, European Federation of Biotechnology. Next one, the principles, now let us start to learn the principles of biotechnology. So from here onwards, lesson important concept will start. Now let us talk. Now principles of biotechnology, among many, so means there are different types of principles of biotechnologies are there among many. So there are two core techniques, ఇది important, there are two core techniques are there that enabled birth of modern biotechnology. So two core techniques so that enables the birth of modern biotechnology. So definitely here there are two core techniques are there. So these two core techniques enables the birth of modern biotechnology. So now what are those? So what are the two core techniques or technologies? Now let us see one by one. So that is first one, genetic engineering. So now in genetic engineering, what is the point? Techniques for what? To alter the chemistry of genetic material. So from here on, what's important? So listen carefully. In a genetic engineering, what is the process we will be doing? Techniques to alter the chemistry of a genetic material. To alter the chemistry of a genetic material. So that is which one and which one? DNA and RNA we can be taken. Why? Because here the genetic material means two types of genetic materials are there now. So DNA and RNA to introduce these into the host organisms. So thus the changes in a phenotype of the host organism. So here the what is the information we are now we are going to discuss is first of all, so we are going to use these techniques or we are going to utilize these techniques. By using of these techniques, so what we are going to do? The changes. So changes you will find there or alter. So we are altering the chemistry. We are altering the chemistry of a genetic material. Chemistry means suppose if you take a chemistry of a genetic material means, so chemistry means here the genetic material is made up of three components. Three components, what are those? So three components are pentose sugar, phosphate and one more is nitrogen base. So in this particular case, no, so we can be changed means when you will take nitrogen bases adenine with thymine, guanosine with cytosine, likewise there are different types of nitrogen bases are there now. All these things means here by changing the sequences, so definitely what we can do by changing or by altering the chemistry of a genetic material and by introducing into the host. So now after changing the chemistry of a genetic material, now we will be introducing into the where? Into the host. So when we will be introducing into the host, what is happening is means, so definitely we can change the phenotype of host organism. Phenotypic means the characters which are expressed out. So means by morphologically we can find out those changes in a host. Means here what is the thing? So one particular genetic sequence is there means that particular genetic sequence we can alter, we can change by changing the chemistry of a genetic sequence. Now by introducing it into the organism, what we can do? So phenotype of the host organism we can be changed. So try to remember. So that particular thing we can be taken under genetic engineering. So genetic engineering, so it is a very important. So genetic engineering is nothing but just simply, so first of all we will change the chemical sequences of or the genetic materials chemical sequence. So after changing of that particular thing, we will be introducing into the host organisms. Then when we will be changing or when we will be introducing of changed genetic material into the new organism, so there in that particular host, we can change the phenotypic characters. If you see this video one or two times, so definitely you will understand the information of genetic engineering. Okay, so genetic engineering. Next one, bioprocess engineering, second one. Now in bioprocess engineering, what is information we will know? The maintenance of sterile ambience. So in this particular case, I have to give some more information to you. Suppose if we would like to do any kind of experiments means, so bioprocess engineering, so maintenance of sterile atmospheric conditions. Ambience means atmosphere, surroundings. So the maintenance of sterile atmospheric conditions in this particular case, what we have to do is, so before start to do any kind of experiments, we have to maintain sterile atmospheric conditions. So in chemical engineering processes, to enable the growth of only desirable microbes. So means in this particular case, at any cost you have to maintain which one? Sterilized conditions. Means suppose in a plant tissue culture techniques concerned, so laminar airflow will be there. So just you try to listen this word and you type in the net and you try to see there. So what is the laminar airflow? So what is the importance of this one? Once you check there. Next one, sterilizations, or there are different types are there. So for example, two I will give you. One is external sterilization and one more is internal sterilization. So but what is the importance of this sterilization means, so we can get means here you see there microbial contamination free. So in brackets for clear understanding purpose I have written, you see there microbial contamination free means, so that particular atmospheric conditions when when it will be very sterile, or when that particular pathogens are not present in that particular state. So in that particular area, suppose if you do any kind of practicals or the experiments also, definitely good result you will get. Okay, but suppose if the area is polluted, or if that particular area like occupied by the microbes or the pathogens in that particular area, suppose if you do any kind of practicals, you will not get success in that particular state. Means that's why even doctors also, when they are going to the operation, so they will maintain one sterile conditions room, they will call it as ICU. So there also, first of all, before entering into that particular hospital rooms, so they will sterilize their hands and with some kind of alcohols. Likewise, so when we will be maintaining that sterile ambience conditions, so then only chance is there for the getting of desirable microbes. So suppose without maintaining of sterile atmospheric conditions, how we can get the growth of only desirable microbes? So all different types of microbes, means desirable microbes as well as unwanted microbes also will grow up when suppose normal conditions are there. Suppose desirable conditions are there, means definitely that enables the growth of desirable microbes and eukaryotic cell in large quantities for the manufacturing of biotechnological products. So for example, antibiotics, vaccines and enzymes. Now, so in order to producing of antibiotics, in order to producing of vaccines, in order to producing of enzymes, at any cost, if you would like to get products in large quantities at any cost, or if you would like to manufacture any kind of biotechnological products at any cost, you have to maintain sterile atmospheric conditions. Then only what is the goal or what is the thing you are trying for, so then only you can get in a sufficient quantities. Otherwise, so that particular area if polluted with different types of microorganisms, you cannot reach your goal in obtaining of antibiotics, vaccines and enzymes. So that's why, so for the first time you have to maintain sterile atmospheric conditions, sterilization is required. So then only, so when you will get like a disease free area or the contaminated area free, so then only chance is there to get suitable conditions, and finally you can get different biological products known as antibiotics, vaccines, enzymes and different types of products. So first of all, principles of biotechnology. So there two core techniques are there. So first one is two core techniques. First one is genetic engineering and the second one is bioprocess engineering. So in genetic engineering, what is the thing we are doing? So by changing the chemistry of genetic sequence or by changing the chemistry of genetic material, so by introducing that changed genetic material into the host, we can change the phenotype of organism or host organism. So try to understand that one. Next one, bioprocess engineering. In this particular case, what is the information we are doing means, just we are maintaining sterile atmospheric conditions in order to obtaining of different types of biotechnological products in large quantities. So try to understand. So this particular chapter, everything, so when you will have interest, so that time only you try to see these videos. Next one. So here let us learn somewhat extra information, that is advantages of which one? Sexual reproduction. So here there are different types of advantages are there of a sexual reproduction over asexual reproduction. Means in our first is in second year studies only, in first chapter we have studied which one? Advances or advantages of sexual reproduction, or in comparison of a sexual reproduction and asexual reproduction, which one is the good means definitely we will say, or which one is better means definitely we will say sexual reproduction. Why? Because now here, why? Because, so reason I have written in once you see there, sexual reproduction provides opportunities for variations. Means suppose if any kind of organisms going to be reproduced by sexual reproduction, definitely so we can see the variations in offsprings. So means here one point I would like to give you. So now even though we have come from our parents 100%. So example, so in sexual reproduction concern, so opportunities for variations means here let me give you one example. So even though we have come from our parents, so 100% without any doubt, but when compared to our parents, so in a color point of view or a height point of view or hair color point of view or either over particular reason, definitely we will be having differences with our parents, isn't it? So means our parents as well as we will not be same, definitely some kind of difference will be there. So that one, why? Because of the sexual reproduction. So this sexual reproduction provides opportunities for variations, means so definitely some kind of changes comes because of the sexual reproduction, but and the formation of unique combinations of genetic setup. Now here definitely, so when compared to our parents, in our body also definitely, so what we can be seen, unique combination of genetic setup we can be seen. Why? Because when our mummy chromosomes and daddy chromosomes comes together, so in that particular case, definitely, so formation of unique combinations of genetic setup we can be observed. You must try to understand this one. So in the first year studies itself, I have given information regarding this one. Actually, sexual reproduction, why here variations comes or variations are there means, so in meiosis, so due to the process of a meiotic division or due to the process of reductional division, so I have given some more information. Why? Because there in which one, in reductional division or in meiosis, so this crossing over process takes place where leptotin, zygotin and pacatin. So third stage, so in the third stage of meiosis 1, so there we have seen, so means crossing over taking place because of the activity of crossing over. So which one, new characters of formation takes place, likewise we have seen now. So that is the main important one. So due to the process of sexual reproduction, definitely what is the thing we can be seen, formation of unique combination of genetic setup we can be seen. Now suppose if you take asexual reproduction, so it preserves the genetic information. Amma, try to understand. So this point already we learned, means asexual reproduction kandipa taking place in within the organism. So not between the organisms. Just in asexual reproduction, what is the point we learned in first chapter? It is this kind of asexual reproduction takes place in single parent. So that's why definitely this asexual reproduction helps only for preservation or preserves the genetic information, but not for the bringing of variations. So this point is an important point regarding sexual reproduction and asexual reproduction. Now let us start to talk about the techniques of genetic engineering. Now, the techniques of genetic engineering, which includes creation of recombinant DNA. So, for the first time only, so they have given very, very important bit, that is, so in a technique, the techniques of genetic engineering means, suppose if any kind of techniques or any kind of processes we are following in genetic engineering, for what means, that gives entire information only one thing, that is creation of recombinant DNA. Sir, first of all, recombinant DNA means so here that information here in these lines you may get. Definitely I will give you explanation regarding that one without skipping this video, try to see. So now recombinant DNA, use of gene cloning and gene transfer. So means first of all techniques of genetic engineering, which includes the creation of recombinant DNA, use of gene cloning and gene transfer. Don't forget this one. Next one, overcome the limitations and allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. So here what is the information I have written now. So now I will explain regarding recombinant gene. So now here, so in first year studies, means in a bacteria, while learning of bacteria, one information I have given, that is in bacteria members plasmid we will find. Emma, did you remember? That is only extra chromosomal semicircular double stranded DNA. Did you remember? So that particular plasmid, so now we will be taking in this particular chapter. So that particular plasmid definitely required for the creation of recombinant gene. So in that particular case now, so in order to creation of recombinant gene, that particular plasmid we will be taking, and by using of restriction enzymes, so we will cut at one place of plasmid. So in that particular place, what is the genetic material we are removing from that plasmid? So in that particular gap, we are keeping our desirable gene, means the gene with good characters, or the wanted gene or wanted characters gene we will be incorporating. Okay, means try to listen carefully. Suppose one plasmid we are taking, extrachromosomal semicircular double stranded DNA, one plasmid we are taking. That particular plasmid we are, by using of restriction enzymes, try to listen carefully, by using of restriction enzymes, just we will cut at one particular place. So there, what is a gene we have cut, that one we will remove, and our desirable gene we will place there. Means when we will insert our desirable gene in that plasmid, now totally in that plasmid how many number of genes are there? 2 are there. One is the gene which is present in the plasmid, and one more is, now what is a gene we have incorporated, isn't it? Now means there 2 different types of genes are there now. So these 2 different types of genes only now combinedly we will be calling as recombinant gene. Don't forget one point, recombinant gene is nothing but the combination of 2 different types of genes together. So suppose if for 2 different types of genes when they are present together, so such kind of particular gene we can be taken as recombinant gene. So I hope you understood this one. Suppose if you are unable to understand, definitely I will explain this entire class in presence of you, no worry. Next one, a foreign gene. Now only I said no desirable gene, or suppose we are incorporating into the plasmid, likewise I said no. So that one more name also is there, that is a foreign gene. A foreign gene which is introduced into the, now you see there, now you will understand very clearly. Now one foreign gene I have taken, so now I am going to introduce into the vector or plasmid. So this plasmid, now I said no, that is only my extra chromosomal genetic material, now I said no. So this plasmid, suppose if you take for biotechnology or genetic engineering processes and techniques, that kind of a plasmid we can be taken as a vector. Don't forget, plasmid we will be using as a vector. Vector is nothing but a vehicle for the incorporation of our foreign gene from the one place to, or from the one organism to another organism. I hope you understood this one. Means a foreign gene which is introduced into the vector or the plasmid, multiply by a specific DNA sequence of a vector or the plasmid is known as origin of replication, or simply ORI. Now just you listen, just a voice only, you try to listen. One foreign gene we have taken. That particular foreign gene also from where we have taken means either one particular organism. We can take any one particular organism there. We have to study about the particular gene sequence of that particular organism. Suppose if you find any kind of desirable gene, or that particular gene may help for the producing of any kind of product. If you find means that particular gene, you will cut from that entire organism, just a desirable gene fragment only you will take. That one now you will incorporate into the vector. How you will incorporate? Now one important point you try to listen. So for the cutting off, for the obtaining of foreign gene, and for the obtaining of a vector, for the obtaining of a foreign gene and for the obtaining of a vector, definitely, so we have to use, or we must use only one kind of restriction enzyme for the obtaining of foreign gene as well as vector. So we should not change the restriction enzyme for the cutting of a foreign gene or the cutting of a vector. So for obtaining of a foreign gene as well as for the cutting of a vector of the plasmid, we must use only one type of restriction enzyme. Don't forget. Okay. So in this particular case, now what I am going to give you is, now this particular foreign gene must be multiplied. Must multiply by a specific gene sequence. Means here this particular gene we are incorporating into the vector or plasmid. No. So why we are incorporating this particular foreign gene must be multiplied. So then on that purpose, now we are depending on the one specific DNA sequence which is present in the plasmid or DNA. That specific DNA sequence only we are calling as ORI, or origin of replication. Means definitely, suppose if you are selecting any kind of a plasmid as a vector, that particular plasmid before select as a vector, that must have origin of replication. Suppose if any vector or any plasmid not having origin of replication, means specific DNA sequence is not there, so that particular plasmid or a vector we should not take for our practicals or the techniques of genetic engineering. Means if you are taking any kind of a plasmid, so for the multiplying of our foreign gene, means at any cost you have to observe the presence of origin of replication. Suppose our origin of replication specific genetic sequence is not there, means we should not take that particular plasmid or vector for our genetic engineering techniques. I hope you understood this one. Means, sir, now suppose if origin of replication is there, then what is the importance of this one? Means I have given already, but one more time I will give you. So this ORI or origin of replication responsible for the initiation of DNA replication or initiation of replication. So means here one foreign gene we are incorporating into the foreign gene, we are incorporating into the plasmid, so that our foreign gene only with the help of origin of replication only that will be replicated. Now thus a foreign gene or alien gene is linked with the origin of replication, so that alien species of DNA can replicate and multiply itself in the host organism. Now here what is the thing? After completion of, after insertion of this alien DNA, where in a particular vector, this entire thing now we are incorporating into the host organism. First of all, one foreign DNA we have taken and one plasmid we have taken. This particular foreign DNA or alien DNA we are incorporating into the vector. Now this particular vector now we are incorporating into the host organism. Now within the host organism, our foreign gene we will be incorporating into the plasmid. So that particular recombinant DNA we will be incorporating into the host. So within the host our foreign gene will be multiplied. That process we are calling as cloning. So that is information now we have studied here. Means here any kind of template or any kind of DNA we can be taken that must have desirable genes. Next one is construction of first recombinant DNA. The construction, now let us see. Construction of first recombinant DNA. Okay, emerged from the possibility of linkage a gene encoding antibiotic resistance with the native plasmid of Salmonella typhimurium. So construction of first recombinant DNA is emerged from the possibility of the linkage of a gene encoding antibiotic resistance with the native plasmid of Salmonella typhimurium. So here this particular phenomenon done by, so this particular phenomenon means for the first time who has been constructed first recombinant DNA, means here we need to learn one point, that is what is the information now we have to learn now. So there the construction of first recombinant DNA emerged from the possibility of linkage of a gene encoding of antibiotic resistance with native plasmid of Salmonella typhimurium. Means in this particular case one particular gene or one particular genetic sequence now we will be taking that must have antibiotic resistance. So that particular gene with antibiotic resistance gene, that particular thing now we will be incorporating into the plasmid. So for the first time this kind of recombinant gene was constructed by Cohen and Boyer, Cohen and Boyer, Boyer. So this one they have accomplished in 1972. So up to here try to save in your minds. So in the next video definitely, so I will come with this continuation. Means here for the first time construction of recombinant DNA is emerged from the possibility of linkage of a gene encoding antibiotic resistance. Means just how we can obtain this recombinant gene, means just one gene sequence we will be taking, that particular gene sequence must have antibiotic resistance, that particular gene now we are incorporating into the plasmid. So definitely at the time now we can take that entire the combination of two different types of genes together we can be taken as recombinant gene. So for the first time they have succeeded in construction of recombinant gene by using of a plasmid of Salmonella typhimurium by Cohen and Boyer in 1972. So this is information regarding just introduction of biotechnology principles and processes. In the next video I will come with a new information. So thank you.